capture. Okay, I think so now it's recording. Okay, so this is going to be a review for the lab practical one. I just, in case you weren't here for the lab when we did the review, or in case you just want to hear it again, I just wanted to make sure I tell you guys what to expect for the lab test. And so remember, it's going to be set up as 32 questions, and it's set up in stations. And so what we see is that the stations are going to be a place where someone would sit, and remember there will be a question there on a note card that looks something like this. And so you'll have your question there. Most of the questions will be multiple choice, but there are some that will say fill in the blank, like for example this one just says fill in the blank and then has the question. And remember, you'll get an answer sheet that has the blanks that you put your answers in. And then some stations will have 2 questions, but most of them will just have 1 question. And the stations will have, for example, a microscope there, and like it could have a question like this one is an example that says identify this part of the microscope, and then in parentheses it says look for the label on the microscope, and then it gives you the options: A, ocular; B, stage; C, nose piece; and D, objective lens. So remember, I'll have a sticky label that will be on the microscope, and so you'll see, like for example, if this was question number 1, then the sticky label would have a number 1 on it, but it will be probably the only sticky label there. But and then you would have to be able to tell me what part of the microscope that is. And then also remember that there will be at the stations for the majority of them that have the microscopes for you to look into. It'll already be set up, so you're not setting anything up, you're just looking into it. Like for example, if I have another example question that I had shown you guys was fill in the blank. What stage of mitosis is shown in this plant cell at the pointer? So that would already be set up in the microscope. Remember, the pointer that's there in the microscope would already be pointing to the chromosomes in the cell, and you'll see lots of cells around it, but you'll just look at where it's at the pointer, and then you'll see that then you'll have to tell me if the cell is for example in prophase, metaphase, anaphase, or telophase. And you remember for that, sorry, I was getting the poster. But remember for that one, for example, if you can see that here, you have your animal cells across the top and the plant cells across the bottom, and I'll have it set up where it has a cell there at the pointer, and you're going to have to tell me if it's prophase, metaphase, anaphase, or like this telophase, or in the plant cell telophase. So remember that 8 of your questions are going to come from that right there. You're going to have a question for each of the phases for the animal cell and each of the phases for the plant cell. So remember that. Okay, and then what else? Remember that you're also going to have to find the letter E. So this is the one question that is your skill question, where I need to know that you actually know how to use the microscope. So remember, we'll have the letter E slide, and you have to put it on the microscope, and then most likely it's going to be bringing it up to high power. I always say it's like low or high power, but definitely if you can bring it up to high power, then I know that for sure you know how to use the microscope. So I would suggest just high power, because that's mostly what I do is high power. But I'll say you have to bring it up to high power, and I'm actually going to be hanging out at that station, so that when there's a person at that station, like for example, when you're at that station, you're going to have to find the letter E, bring it into focus, and I'm going to look in there and then say, yep, you did it, and on your answer sheet I'll initial that yes, you did it, and that you know how to use the microscope. What's some other examples? Okay, so for example, this was another question that I gave where it says identify this organelle in the animal cell model, look for the label on the model. So see, we have our animal cell model, and what you want to do is, I had recommended taking a picture of this, but basically you want to compare this to your picture that's in your lab manual, and you're going to have to identify organelles on here. And so just for example, we have the nucleus here, and it has the nucleolus in the middle, and then we have centrioles in the centrosome. All of this, that's that green and blue, is going to be endoplasmic reticulum, and I don't normally distinguish between the rough and the smooth ER, but just in case, the areas where it's showing there's just the blue, that would be smooth ER, but then where it has the white dots on it, that's going to be the rough ER. This right here is going to be the Golgi that has little vesicles. And then we have our mitochondria, which you can see the inside, or sometimes you can't, but they're orange. And then lysosomes and peroxisomes, I don't normally ask about those two, just because they look very similar on the model except for their color. So I don't ask about those, just so you know. But those are the organelles that I could ask. I'll have a little sticky label on one of them that has the number, and this one could have 1 or 2 that you have to identify. And so you would just look for the sticky label and then tell me what it is. And for the most part, I put those as multiple choice for you. And it'll be the same thing on the plant cell. So let me hold this up a second. So on the plant cell, see, we have our nucleus and nucleolus. We have smooth ER, rough ER. And we have Golgi apparatus is going to be up here with some vesicles. And we have mitochondria. And then we, let's see what else. Oh, and then we see the chloroplast look like this, or like this, or this. And then we all have this really large organelle, remember the central vacuole. So I'll ask for any of those. And then also, when you're studying about the microscope, also remember I said one of the example questions I gave you, you're not only going to have to know the parts of the microscope and how to find like the letter E on there, but also, this is an example. It says fill in the blank. The ocular lens is 10x, so what is the total magnification when using the scanning power? And so remember, I'm telling you the ocular lens is 10x, and then we have our 3 objective lenses. We have scanning, low, and high. And people get scanning and low confused all the time, so just make sure you know. But remember, to figure out the total magnification, you multiply across. So in this example, it says scanning power. So you would do 10 times 4, which I'll have the microscopes there where you can look at the objective lenses and you can see that on the objective lenses, remember, it has the numbers 4, 10, 40. And you just multiply by 10, the objective lens. So 4 times 10 would be 40, so that would be the total magnification. And that one's a fill in the blank, so there's not the options there. I want to make sure that you do know how to do that. That's really important. Let's see, what else would I ask? I am going to ask about meiosis. And remember, in meiosis I had my example cards. These were when I did the demo of the meiosis slides that were set up. But let me grab my poster. Oh, okay. So here's our poster, and see across the top it's got prophase 1 all the way through telophase 1. And so let me remember that with telophase 1 there's going to be 2 daughter cells. And then let's go down here. Here's our meiosis 2. Let me get this straight. Okay, sorry. In our meiosis 2 we're going to have at the beginning of, whoops, I got it all wonky again. Let me just, I'm trying to look over here though. Hold on, let me okay. 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 Sorry. So we have our prophase 2 is going to start out with 2 daughter cells. And then by the end of meiosis 2, by the end of meiosis, the whole process, remember we're going to have our 4 daughter cells. So that's a big clue. So Remember, I'm going to have the different ones, like early prophase, beginning of meiosis 1, the chromosomes are stained pink, they are condensed. During meiosis 1, the chromosomes are on metaphase, lined up on the metaphase plate. End of meiosis 1 or beginning of meiosis 2, you can see there's 2 daughter cells. And then end of meiosis, you can see there's 4 daughter cells. So I could have any of those on there and set up for you and ask you questions about that, where you have to be able to recognize maybe when it happened in meiosis, or why, you know, like, is it 2 daughter cells, 4 daughter cells? And then let me grab this real quick. Okay, so then also remember, you want to study your lab exercises. So you want to go through, and anywhere that there's bolded words, you know, make sure you know what they mean. And then just go through, like, you had to know what's going on with each of the stages of the cell cycle, and you had to know the parts of the chromosome. There's all, just go back, look through here. It goes through, has the pictures showing us what's happening in mitosis, and it also has the statements underneath telling us what happens in mitosis. You know, you need to know that. You need to be able to recognize, you know, cytokinesis in the animal cell and the plant cell. So if I have an animal cell or a plant cell on there and I ask you, like, what's this structure seen in the animal cell at the end of mitosis? Well, you better know it's a cleavage furrow, because it's at the end of mitosis, and I'll have the pointer pointing at it. Or if it's the plant cell, then you better know it's the cell plate. Okay, so that kind of stuff will be on there. And then also, you know, when you're going through and studying, just remember that you're studying the review questions as well. Because sometimes, remember I told you when we did the review in lab that there are times that I have just regular questions that are on the test. You know, that I might have gotten from the review questions, or I might have gotten them from somewhere else. But, you know, for example, let me see if I can find an example of one, and I'll just tell you an example. If I can find it. If I can find one, that's an example. Just give me a second. I know I can, I'm just... Oh, here's one. The nuclear division that results in 2 new nuclei, each having the same number of chromosomes as the original cell, is called, and I have A, mitosis, B, meiosis, and C, the cell cycle, or D, duplication. So see, that's just a regular one. There's no slide or microscope there. Fill in the blank. Blank cells are produced in the ovary. That's just a regular question, no slides or anything. So I do have regular questions like that on the, I call them regular questions, meaning that they're not like necessarily set up with anything at the station. There's just a question there. I think that's it for, you know, how to study and all that. Trying to think what else there could be. But I did want to make sure I tell you that, you know, remind you that the test, we all start at the same time. It's a timed test. So remember, the door will be locked until the test is supposed to start. And if you're in my Monday, Wednesday, Friday class, that's going to be Wednesday at 1 o'clock. And if you're in my Tuesday, Thursday class, we're going to have it at 9 o'clock. And you can't be late, because we're all going to start at the same time and we're going to finish at the same time. You don't want to walk in the middle of the test and we're already halfway through it. And when you come in, what you need is, all you need is a writing utensil. I would suggest a pencil in case you have to erase your answer and put a different answer. And I'll give you an answer sheet to put your answers on. And then if you bring a bag or books or anything with you, you'll just set them at the back of the lab like by the countertops, by the door, by the lab door. And then remember that you can start at any number that you want, or any open station. So if you start at number 12, for example, you'll just make sure that on your answer sheet you're on number 12 answer. And then you'll just, when I say switch stations, you'll go to the next station, number 13. And you just need to make sure that you check that, so that when you are at your next one, that you did go the right way, and so you don't get off. And then remember that you'll get a minute per station. And it doesn't sound like a lot of time, but most students honestly don't use the full minute, but you're welcome to use the full minute. Some do, or for certain questions you might need to use the full minute, but not for others. And so it's basically you either know the answer or you don't. And actually a minute is being generous. I remember I told you that in most other courses with lab practicals, they're just all fill in the blank, and they're all, so remember mine are not all fill in the blank, but they're all fill in the blank, and you get like 20 to 30 seconds. So I am being generous, even though it seems like, oh my gosh, that's not very much time. But you either know it or you don't know it, okay? And then when the minute is up, or if I see that everybody's finished answering, because you'll close up your answer sheet and you'll just look forward, then I'll say switch stations, and you'll go to the next station. And when you so like, let's say if you started at number 12, by the end of the test your last one then would be number 11. Okay, so you'll eventually end up going around answering all the questions, and we all get done at the same time. You can't go back and look at any of the answers or questions, or you can't go back and look at any of the questions to change your answers or anything, or to think about them longer. When we're done, you turn it in. But then, and then you'll sign the attendance sheet after you turn it in. Okay? So please let me know if you have any questions, and I'll be happy to answer your questions or go over anything with you again. But thanks.